DNA replication. It's an important process in our body because it replicates our DNA and helps us grow, repair, and fix our cells. Let's first start off by reviewing what DNA is. DNA stands for deoxyribonucleic acid. And in this, shown in this example, is a DNA strand going under DNA replication. This is the replication fork, where helicase, an enzyme, unzips this DNA. Here we can label it as replication fork. This will be helicase. As shown here, as these DNA strands are being unwound, these become the coding template strands, and here new DNA is being added in.
In order to add and create the new DNA strands, there needs to be free nucleotides as shown here, here, and here, that DNA polymerase can add free nucleotides.
However, even before DNA polymerase can start adding these free nucleotides to the coding template strands, we first need a primer called DNA primase, which will start and allow DNA polymerase to attach on and to start synthesizing the DNA.
Here is DNA primase. After DNA primase is laid down, DNA polymerase can attach to the template strand and start creating a new strand.
Quick note, when helicase is unwinding the strands, there is also another enzyme that counteracts this to prevent DNA from tangling up after it's being replicated. This is called topoisomerase. This helps prevent further tangling of the original DNA strand as helicase is continuing to unwind the strand for the DNA template strand to create more nucleotides to add.
It does this by snipping the DNA in parts so that it wouldn't get tangled. This is two pieces of string that have been wound together. So if helicase were to just split the DNA strand open like this, you would see how the DNA in the beginning will get tighter and tighter and tighter and start to tangle. And at one point, helicase won't be able to separate the strands anymore because it's too tightly wound. So it is topoisomerase's job to prevent this from happening so that the full DNA strand can be unwound.
When DNA polymerase is adding new nucleotides to the template strand, it must follow a specific rule. This rule is consisted of these four different colors shown here. In this example, red represents adenine, yellow represents thymine, blue represents guanine, and green represents cytosine. As the diagram already shows, adenine can only pair with thymine, while guanine can only pair with cytosine. This is because the specific shapes of each of these base pairs only can match with the others. In fact, adenine and guanine are both considered purines. Being a purine means that it is consisted of two rings. Thymine and cytosine, on the other hand, are pyrimidines, which means that their structure only has one ring. Thus, for example, with adenine and thymine pairing together, this means that adenine with two rings and thymine with one ring can in total make a width of 3 rings in total, which allows for the DNA strand, this double helix strand, to be consistently the same width throughout.
Because purines are longer than pyrimidines, if they were to pair together, it would create a section in the DNA that's wider than if two pyrimidines were to pair together. This creates inconsistency in the width of the DNA strand. Thus, it is why purines pair with pyrimidines. Okay. When DNA is unzipped, there's going to be a 3' end and a 5' end. DNA polymerase can only add free nucleotides to the 3' end. This creates two different types of strands. The first is the leading strand, and the second is the lagging strand. It's called the lagging strand because DNA needs to be created in fragments. These are called Okazaki fragments, and they also have a 3' and 5' end.
Let us not also forget that there is also a primase here. And DNA polymerase is also adding new nucleotides to this, except in this direction. With the leading strand, this is a lot more simple because this can easily follow this direction.
For the lagging strand, it needs to be created in small fragments, and these fragments are later joined by a different molecule called ligase.
To join this, we have another enzyme called ligase. This enzyme works like the glue of DNA synthesis. It brings all these fragments, especially these Okazaki fragments, together,
creating one seamless DNA strand. So as you can see that there are sections here where it's still just a primer, but you can't have primers disrupting your DNA strand. What do we do about that? Well, there's another molecule called DNA polymerase 1. And DNA polymerase 1 comes in after DNA polymerase 2 has already finished its job, and it replaces the primers with the proper nucleotides needed to create the DNA strand.
But before this whole process is finished, let us not forget about the enzyme that's working behind the scenes of this entire process. These are called single-strand binding proteins. These exist around this part of it. They stabilize the unwound parental DNA. As this diagram shows, DNA replication is a semi-conservative process. You can see that the parental DNA is being preserved in both new replicated DNA. Half of the parental DNA is being preserved, while the other half consists of new DNA being added by DNA polymerase 2, adding the new nucleotides. To make a bigger picture of DNA replication, we can see DNA replication occurring in all parts of life, from mitosis to meiosis. In fact, DNA is the blueprint of life.
It is what allows us to pass our genetic information on into the future generations.
Thank you, DNA replication, for making life continue.
That's a D!
That's a D! Can we not?
Learning as we go.
Just like, what have I taught you guys?
A quick note to this. What is English?
Can we please ask?
Yeah.
Okay, there we go. I can't even.
We're doing that. We're using that. Okay.